So we can actually compartmentalize that last finding just to right here. If you make your two gametes, you make your sperm and you make your egg, and you get them together, that's fertilization. That gives you a new critter. Okay? So we study meiosis because this is the process we need to reduce our genetic components from 2N to 1N. If you put 2N together with 2N, you're going to end up with 4N. Put 4N together with 4N, you're going to get 8N, and so forth. The process is only supposed to give us recombination of the different genes, not to amplify them. So this is the way to systematically reduce and then re-amplify the amount of DNA that a cell carries, so that when you make a new person, you only have the regular complement of stuff. These are central principles to meiosis here. You can't take a haploid cell and divide its DNA content into two and expect anything systematic to happen. So we do this with diploid cells, again from 2N to 1N, and we always end up with haploid cells. Okay? And I think that's move on. So this is just the nuts and bolts, a simple list of the stuff that happens in meiosis. Okay? It's essentially 2 rounds of mitosis with one really big twist. Okay? So we're going to undergo 1 round of DNA synthesis before any of this begins. So we're starting as 2N, but 2N replicated. We'll then enter the first division and its whole process. So prophase 1, just like mitosis 1, just like mitosis, is going to organize the chromosomes, but with an important difference. Prophase 1, this is the singular most important thing about meiosis that's going to trip you up. This most important difference right here. Prophase 1 is where homologous chromosomes line up. They actually are physically next to each other such that they can cross over each other, literally. And when they do that, it creates breaks in the DNA, either enzymatically or by accident. The mechanism is not entirely clear. But when they do that and they cross over many times over each other, those 2 pieces of DNA, one from homologous chromosome A, one from homologous chromosome B, actually swap the sequences of DNA on those chromosomes. So hopefully if we come back to this and you think about this, you could say, do I have my grandpa's chromosome or my grandma's chromosome? And the answer should be neither. When however, your parents would. Say you're talking about your paternal side. Your dad would have a chromosome from grandma, you would have a chromosome from grandpa, whichever one I didn't say. But when he went through the process of making sperm to make you, those chromosomes completely blended together, and now they are completely distinct from anything that he inherited from his parents. So this is enormously important for amplifying genetic diversity that's valuable to a species. So you've got a novel trait that comes by mutation. If you can't redistribute it to other chromosomes, it's not going to spread through the population very efficiently. So that's its impact on the species. That's all downstream knowledge of this. But I hope you can sense by how much time I spent on this. Prophase 1, important because it has crossing over. On the count of 3, when does crossing over happen? 1, 2, 3.
Prophase 1.
Great. Metaphase 1, again, we're lining up our different genetic materials along the center of the cell. But importantly, what we're grabbing a hold of is not sister chromatids to pull them apart, but homologous chromosomes to separate them into different cells. So anaphase 1 will then separate those homologous chromosomes, again destined for unique cells here, and we'll divide them into nuclei in telophase 1 and divide the cells in cytokinesis 1. Meiosis, I should point out at this point, but first let's take a public opinion poll. At this point, are these 2 daughter cells haploid or diploid on the count of 3? 1, 2, 3. See, it's tricky.
They are haploid. We have separated each homologous chromosome pair, and only one homologous chromosome from the original cell is present in each of these cells. It is, however, replicated. But remember, when we talk about ploidy, haploid or diploid, we're talking about the number of chromosomes, not chromatids. So while for each chromosome that you have on here, you do have 2 chromatids, each chromosome is unique. If this is a human cell, you would have 23 unique chromosomes on this, each of which is replicated. So on the count of 3, are these cells haploid or diploid? 1, 2, 3. Haploid. They are 1N, what I would call asterisk, because they are replicated. So after we have each of these cells, we go through meiosis 2, which is, for all intents and purposes, identical to mitosis. One important thing to consider, we do not go through another round of DNA synthesis. There is no S phase. And so therefore we go from 1N asterisk, so 1N replicated, to 1N total. Okay? So each of these cells, they'll reorganize their DNA during prophase. They'll line them up during metaphase 2. We'll separate the sister chromatids in anaphase 2. Now I'll point out that this is another one of the wrinkles. In anaphase 2, when you're still in the same cell, when you don't have any nuclei, the number of chromosomes actually doubles after anaphase 2 is complete. So we were 1N asterisk. Right now we are 2N because we split the chromatids. But we will very quickly parse those into one set each into the descendant cells. Once you've gone through this 2 or 3 times, pay attention to this little factoid. Because of the crossing over that happened in prophase 1, these copies are not actually identical, okay? But the way that you'll see the models, they'll be represented as mostly identical. So you need to be wary of what we're trying to get through to you at each phase of the instruction. So in the simple case, each of these cells would look pretty identical to each other, but because of crossing over, you'll notice that little arms here and there are different on each of the per each chromosome. Okay, cytokinesis 4 gives us 4 haploid cells at the end. So that's nuts and bolts. So this is really just a chance to reiterate, although you will see down here the beginnings of that fine point that I just made, that some of these chromosomes are actually slightly different from the others. So important differences. First, you have prophase 1. The homologous chromosomes are organized into tetrads. Homologous chromosomes actually find each other out. How they do it, I don't know. I think it must be pretty magical. But they get together. There are 4 copies of the same genetic material there. 2 copies each of the unique chromosomes in the cell. At that point, you can have crossing over, the genetic exchange between homologous chromosomes. You then separate those homologous chromosomes through the process of meiosis 1. And then meiosis 2 looks and feels pretty much like mitosis, except the resulting cells in meiosis are going to be haploid. And again, that's because there is no second round of DNA synthesis for meiosis. Yeah, and the only other thing that I noted on there is that there is 2 rounds of cell division rather than 1. I think that's pretty obvious, but worth pointing out. If I can take the moment to try to illustrate what crossing over results in, look at up here. We've represented a cell with 3 unique sets of homologous chromosomes. Okay, so we got 2 copies of each, and when we line up, there's a red copy of the short one, a blue copy of the short one, red medium, red large, blue large. Okay, during this process, the tips will switch sides, and you'll end up with, they're putting it in metaphase 1, I don't buy it, but you can see they actually draw them crossing over there. And as a result, you end up with something that one of the sister chromatids on one of these chromosomes, after anaphase separates them, has this little blue tip from the one over here.
Okay, I hope that's a little bit wild for you, but as you look at this, make sure you notice that everything that's got a change in color has the corresponding change on the other matching homologous chromosome. And so these cells, now reduced to haploid but replicated, when they go through the second round of meiosis, they're going to be splitting down the middle here, such that you'll see something like one cell will inherit this entirely red half, one cell will inherit the recombined one, and the complement will be in these cells over here, which were descended from the complementary chromosomes. So please examine that. I won't pretend like that's the best. Here it is again. I think I'm going to spare you re-explaining it. But I'll just point out that in my scheme, replicated DNA, which is not a measure of how haploid or diploid it is, I represent with an asterisk, so I can keep track of it. So the first phase of meiosis splits up homologous chromosomes. That's why we end up with 2 haploid cells, but containing replicated DNA, and they get distributed to 4 very typically haploid cells. When you do your bead models, this is what the crossing over will look like. Make sure that you accomplish this goal in your exercise, because this is the sort of thing that helps you know that you understand it and that shows up on tests. So then we've got a couple of anatomy items, and so it's the 2 reproductive tissues of the testes and the ovaries, where we're going to be making our gametes. Okay, so in terms of the gross anatomy, we're only going to point to 2 different things: the seminiferous tubules, that's where all the proliferation is happening, to proliferation and meiosis, I should say. So that's going to generate the cells, but then they're going to go to the epididymis to mature and be stored. Okay? And the main thing about these one of the things that we really highlight about the different gonads is their role in hormone signaling. And so the interstitial cells, which honestly we don't really ever actually look at, we just kind of know that they're in here. Those are responsible for the release of testosterone, which gives us the male secondary sex characteristics. And there it is. We'll have the complementary hormones when we get to the ovary. So the big thing that we want you to observe is how, let's put it this way, the major form meets function point on the testis is that it works from the outside in terms of maturing. So on the outside of the seminiferous tubule, you have the most immature cells. On the inside, you have the most mature. And when you look at this under the microscope, that's the kind of organization that you have to figure out. And the things you're looking for, sorry, one more point before I describe that. The terminology that goes with this is obnoxiously just have to memorize it and somehow have to get it straight in your mind. Okay? From the outside, the most primitive cells, the ones that are actually diploid, the ones that self-renew through mitosis, are the spermatogonium. And so we have to pay attention to the last couple syllables on each of these terms. The most primitive is the gonium. Same holds true for making eggs. Those are the oogonium. Once we have actually entered meiosis, and you can see this by them going into prophase, they're just 1 or 2 cells deep into the tissue. And that's something you should observe when you look at these things. You can see that they've entered meiosis. They are therefore a primary spermatocyte. Once they have completed a round of meiosis,
and they're going into the second, they're then a secondary spermatocyte. Okay, and the salient feature there, look,
it's even on the slide, is that they're haploid. Okay?
After that, we'll complete that cell division, and you'll see that there's a bunch of cells who are busy losing most of their actual cytoplasm, and they're basically just condensing down into one big ball of wiggling DNA. So you see very heavy nuclear staining at the inside of the seminiferous tubule. That head is of a maturing spermatid. And even at spermatid, it's still not a mature sperm cell. Spermatids will grow their flagella out the back so that they become motile. And once they finally do that, we can call them a spermatozoa, or a sperm. So when you're going through this, thankfully the eggs have a similar terminology, but you do have to memorize that order from outside in. I think I put a better figure in here. I like this one much better. So this is the actual tissue that you'll be looking at.
Again, make sure you look at the layers and know what's going on from outside in. So on the very outside we have the self-renewing mitotic cells. This is how men can make millions of sperm every day. They mature. You can see there's an actual change in their morphology, just one cell layer in. Sometimes it's 2 cell layers. That's a primary spermatocyte. These are various levels of maturation through the meiosis process.
It's very hard to discern anything there. And these cell divisions are very quick, so we won't be assessing whether or not you can tell is it primary spermatocyte or secondary spermatocyte. But when you get down to the inside, you can see that the nuclei are very condensed. Those are the spermatids. You can see that they're growing their flagella, and those are actually streaming and waving into the lumen of the seminiferous tubules. Once those are mature enough, they'll be released from the tissue and then go get stored in the epididymis, where they actually mature over a course of months. So then when we talk about the egg, I'll tell you right now, the number one thing you're looking at for the eggs as they mature, and it's this process of maturation that's central to what we're going to be testing you on, okay? So that's what you need to work on. Looking at the egg itself will not tell you anything. The only way to distinguish these guys from one stage from the other is their surrounding follicle cells, or rather follicle tissues. So when we first start out, let's point out that women, there's some contention, but it's pretty generally accepted. Women start with all of their primary oocytes before they're born. So the whole process of initiating meiosis happens while you're still in the womb. You do not generate further eggs. So there is no analogous structure to the spermatogonium in humans. If you're a rabbit, it's different. You're going to be continuously renewing these things. But it's something we do different. Okay, when we look at us, the primary oocytes, it's a big pink cell with a single squamous epithelial cell around it. They're actually kind of hard to find, okay? But they also do you this favor of working outside to in. So take a look at the spatial arrangements when you're looking at the tissue. After that, you'll see the epithelium around it swell. So it'll be 3, 4, 5, 6 cell layers thick. And the follicle cells, they tend to stain very nice deep purple on the tissues that we're going to show you. Okay, so that's the primary follicle. Before it swells, it's called the primitive follicle. And as it moves on to a secondary follicle, so this is where you actually, I believe the stages actually match the terms. So secondary oocyte should have been through one round of meiosis. Yeah, there it is. So once we are now haploid, you have a secondary oocyte. Again, you can't tell much about it by looking at the oocyte itself, but the surrounding follicle will start to have these perforations. That is, it'll start to open up spaces in that thick layer of follicle cells. Okay? It is There's a little bit of quibbling about the terminology, should there be a tertiary stage or not. In this class we skip tertiary, and we just say that once all of these perforations, you can see they're growing in this representation,
or you could if it was closer to your screen, or you were closer to the screen. Once those are all connected and you have a nice big lumen here,
that is a mature or Graafian follicle. And that oocyte, though still containing replicated DNA, is ready for its release. So during ovulation, this whole chamber bursts and the egg is released down the fallopian tubes. I think it varies by species, but ultimately the cell will go through its final meiotic division and become mature. I think one of the contentions is that doesn't actually even happen until a sperm gets into the cell. So we're not testing you on that, but that rupture, that is ovulation. And let's see. So this whole vesicle here will ultimately compositionally change and become yellow due to some storing of some beta carotenes, and that will form the corpus luteum. Luteum is Latin for yellow. Okay, we'll see it again when we look at the eye, believe it or not. So the corpus luteum is there essentially for about 2 weeks once the egg has been released, and its major job is to release progesterone, which is the hormone that promotes implantation upon fertilization. If fertilization happens, there will be feedback from another hormone, HGC, that will keep the corpus luteum active and producing progesterone. And so that will maintain the pregnancy. If it fails, the corpus luteum will just kind of go away, and therefore it will no longer be releasing progesterone. And the other major activity, the other major hormone activity,
that is production of estrogen, which promotes the menstrual cycle, will continue to be produced by all these follicle cells along the way. So estrogen in the follicle cells, progesterone in the corpus luteum, and you can take your pick however you want to make your birth control. They're both equally viable options for that. You can either convince yourself you're pregnant, or you can convince yourself to never become pregnant. Both work for not having new babies. One final thing before we look at the actual histology, that unlike the meiotic divisions in sperm, the meiotic divisions in oocytes produce unequally sized cells. So the purpose of making an egg is something that can actually go through the first rounds of cell division for the zygote. So it's got to have lots of cellular resources stored up, everything you need to make a very young person. So the actual oocyte that's done is the biggest cell in your body ever. It does that by spinning off essentially only the DNA, and those little chunks are called polar bodies. And so there's, I don't know if this actually goes through the second round of division, but there's 2 sets of polar body generation where we're just spinning off excess DNA. Finally, here's what it looks like. So we don't actually have a primary follicle on here, or primary primitive follicle, or the oogonium, or the primary oocyte. But these large pink cells are what you're going to be looking for in the microscope. You can see here that we've actually got a decent sized follicle already, but it's actually starting to perforate. So this is getting towards secondary oocyte territory, secondary follicle status. This is further along. I would still call that secondary, but this is essentially mature. You got a huge cavity and a slightly surrounded central cell here.